Hello, welcome to this video. I'm Nicole. So in this part or third part, we actually going to do the actual karyotyping in this video. So what will happen is I'm gonna cut the chromosomes and then I'm gonna stick them here onto this karyogram. So once the blood, bone marrow, or amniotic fluid or CV is cultured in the lab, like the harvesting and staining, all the processes are done, they get scanned, or you can just use the slide and view under the microscope. So under the microscope, you're gonna see something like this. This is your metaphase. You're going to see the chromosomes that are nicely spread. If you did respect the volume of calcimide and the time and everything, your chromosome will not be too short and all of that. So you're going to see something like that. This is amnio. So it looks a little bit like you can see. This is also amnio. I don't really have much blood, but I can tell you that bone marrow, it's going to look much better than culture, amniotic fluid culture, and the blood will look way better. It's gonna be so big. I just don't know why I lost sight in terms of making copies of how bone marrow and blood looks like. But this is how amnio that I had looks like. It is also amnio. So you're going to have a metaphase, or many metaphases. One of them may or will look like this. Chromosomes are nicely spread. So you are being asked to do karyotyping. So basically, you are being asked to recognize your chromosome, to cut them, and to paste them or stick them onto this karyogram. So if you look at this metaphase, you think that, which is actually true, that this is your chromosome 1, this is your other chromosome 1. You're gonna cut your chromosome 1, you cut your second chromosome 1, and then you come here onto the right spot. You're going to paste your chromosome 1s. So you cut them over there, you paste them here, and this is how you do your karyotyping. So you're gonna paste them two correctly, and then at the end you are going to write the ISCN number here, which is going to be the number of your chromosome. Let's say it's 46. You're gonna write here 46. If it's a female, you're gonna write XX. If it's a male, you're gonna write XY. Y. If there's any abnormality, you will also add them here. So this is basically it. So karyotyping, it's a genetic test. It's a test that is done in the cytogenetic lab, where it's like a snapshot of human genes, of all the chromosomes. So we're going to spread the chromosome here. We're going to order them. We're going to place them in order so that we can look and see if there's any changes, if there's a change, any structural changes, any change with the structure or with the number. Because when those changes occur, it does result in abnormality. So if there's any changes on the structure or the number, instead of 2 sets of chromosome, we're actually having like 3 or 1. So that's rearrangement. Is definitely telling us that there's something wrong. So from that we can deduct what abnormality we are dealing with, what problem the patient has, and take it further. So I hope you have an idea what are the steps of karyotyping. So you get your metaphase, you cut them. You have the choice of labeling them like this. You can name them or number them, give them numbers before, then you cut them after, then you just paste them here. Or you can just cut them when you're done, you paste. Either way it's fine. Just make sure you know your chromosome, what is what, and where to place them. And if there's any other thing that has been rearranged, whether it's in the structure, in the number, any changes at all in the structure, in the number, you need to take note of that, because those changes, those rearrangements, it's where lies the problem. It's where lies the abnormality or the disease that we are looking forward to. Not looking forward to, but we aim to find or detect as a cytogeneticist. So yeah. So what I'm gonna do, I'll pick one of these. I'm gonna cut them right now. And then, yeah. I'm not so sure if I should label them with you. Let me just show you how you can label them. Let's see if I have red pen. So you have the choice. Oh, did you watch this video where we actually drew all this chromosome and explain? So you have the choice to look at it like this. Like when I look at it like this, normally we have this when we do karyotyping for each and every metaphase. Just give me a second. We have this sheet that helps a lot. So whenever, like here, I already wrote chromosome 1, I will just come here onto these sheets and then I write like 1. If I find another chromosome 1 as well, I will come also tick here 1. So this is really good if you're going to use this spreadsheet, because it's gonna help you keep track of the chromosomes. So if you find a chromosome that looks like another chromosome you're thinking, you just come here and say, oh, I already have my 12, so that's definitely is not my 11. Or what I'm thinking is my 11 is actually my 12. So this sheet really helps. You look at your chromosome 1, 2, you write here, you make sure you got them right. So it's also gonna help you to prevent any you from saying there's a trisomy when there's not a trisomy. You just counted the wrong chromosome as the third chromosome of another chromosome. That's how you end up with a false chromosome, which is a crime in the cytogenetic lab because it's not tolerated. So some people do it like from chromosome 1 to 46, but me, I don't know, for some reasons, when I look at my metaphase, like this beautiful metaphase from amniotic fluid cultures, I just look at it, then I start numbering them. So when I look at this one, I think I already did some. I will just go and write, this is my 16. This is definitely my 17. This is my 21. This is my 18. This is my other 17. This is my 9. This is my X. This this is my, looks like my 19. This is my other 16. This is my definitely my X. This is my 15. So this is my 19. This is my other 19. So this is my 20. You see this dark band here. So I like doing this because it goes fast for me. I can do this like 15 20 minutes. All my chromosomes I have number. But for me not to confuse, I will have to rely on this. So whenever I point like this is my X, I have to come here and tick X just to make sure that I don't end up counting one chromosome more than I should. You see? Yeah. But the other way, you can just cut directly and paste them here. Yeah, without numbering them first. Or you can number them first and paste them here. Do you understand? So yeah, I usually do this and then I paste them. So right now, I'm not so sure if you want me to cut this one or to cut a bigger one, because the bigger one will be more visible than this one. Either way, I'm gonna cut and paste them here. So at the end, this is how they're going to look like in pairs. They're gonna look like this. Then we can count the number and check if there's any deletion, translocation, any chromosome abnormality. Okay. So I think I would have loved to cut this one, but I'm just concerned because the size is pretty small and I'm almost like done. So I'll cut this one and then I will just mix them, then I'll paste them here. So you're gonna watch me right now doing the cutting. Okay, shall we? So while I'm cutting, I don't know if I should talk to you or put just some music. So yeah, I think you're gonna get there. You're gonna get there. It's just a matter of practicing. So I cut these two. Let me just put them all together, because I don't want you to say, oh no, they're already set. That's how you were able to find these chromosomes. So usually after cutting, I will just, paste them. I will just stick, oh yeah, but today I'm just going to what am I going to do? I'm gonna mix them together so you can see how we can actually find our chromosome. Do you want me to keep speaking or put some music for you? Let me put some music for you. Maybe you are tired of my voice by now. Oh my gosh. If you were not tired of my voice, I could have been able to. So yeah, I picked this one because first of all, it's bigger and you can clearly see, and another thing is it's gonna go faster. You see, if I pick those ones, the amnio, it's going to take a while to cut them because the chromosomes are pretty small. But these ones, the chromosomes are pretty big. So it's not gonna take me so much time because yeah, I have 3 videos to shoot today, so it has taken me so much time already. And then, as you know, I'm a very busy woman. I have a very busy schedule, so I can't afford anything that takes me more time than I can give. So this is going fast, and I'm so happy that we're gonna finish. Ooh, that's something on the outside that we're gonna finish pretty soon. Once we finish, I'm definitely out of here. So karyotyping is not difficult. I thought it was difficult. I thought it was the end of the world. I thought I was not never gonna know the chromosome. I thought it was impossible to know them. I thought so many things, I freak out. I'm like, how am I gonna do this? At some point I was like, why did I pick cytogenetics? I don't know this chromosome. What is going on? But then, at some point, I just remember the advice that was given to me. She said to me, it's gonna come with practice. You just have to practice. You just have to do it. So I kept on practicing. Today we are here. I don't know if tomorrow. You guys need to let me know, hey? Let me know if you like to see other videos, maybe on how to do karyotyping for trisomies, other aneuploidies, whatever cytogenetics or medical laboratory science, lab science you would like to see. You let me know. as I'm speaking to you. There will be also a video on how to make your agarose gel electrophoresis, PCR. Please, if you're interested into molecular, you can also watch that video. Also, we do have a video of a day in my life as a medical lab scientist in this channel. If you haven't watched it yet, you can check it out. More importantly, subscribe, comments, like, join the family, show support. Don't be a stranger on this side, watching in silence. Show support, join the family by clicking on the subscription button, and by clicking on the notification bell so you don't get to miss any video, as I choose to upload a video every single week. So yeah, there are so many videos in this channel, so it's very accommodating. Whatever life, you know, background you have, your gender, you should find a video that does accommodate you. Definitely. There are so many videos that you're going to definitely enjoy watching in this channel every day. We are working hard into improving, into making you happy, my precious subscriber. So in this world of improvement, decision of improvement and practice and commitments that I made myself for my subscriber, my most precious one. There are not so many, just little. That's how they are so precious, because the number is just so small, makes them special and precious. Committed to uploading videos every single day, every single week. What time am I gonna go to the live if I have to post every single day? And then reading, you have to read, you have to research, you have to improve yourself, get all the knowledge you need. So we have a set of chromosome here. To avoid any form of cheating, I'm gonna mix them again so you can be sure that I don't know what is what until I lift them. The next thing you're gonna need in your white pencil case here and open, like, it's okay. The next thing you're gonna need is this. So you're gonna place them here. As I'm doing so, I'll just keep chatting to you, because you know, chatting to you is therapeutic. You know, in most of my vlog, I just say, you'll be surprised how therapeutic it is to watch me dance. If you haven't yet, you must watch me dance. Check my other videos. It's therapeutic to watch me eat. It's fun and relaxing. So back to this. I think with practice, you will get here. You will really get it. Like, yeah. It is no magic. It is no magic. It's just about practice. So, yeah. The more you practice, the more you make it. So these are my chromosomes. I will start putting them where they belong. Normally, yeah. No, it's fine. This, I'm not so sure, but they look like my 5. So if you are not so sure, you can just put it on the side. There's nothing wrong about not being sure. But when I look at this one, I'm so sure that this is my 9. So I'll take the thing and then I put it here. So when I look at this one, I'm so sure that this is my 1. See? I just put this guy here. I look at this one, I'm like, oh my gosh, this is my pretty little 4, you see. So I just pick it like this. My nails are long. When I do karyotyping with my long nails, sometimes life become a little bit difficult. So when I look at this one, I have a vibe that this is my 2. But yeah. So I'm just gonna put it here. If for some reasons I'm not happy, I will just change them. But you have to be sure. This looks like my 12. It actually is my 12. I'm gonna just put it here. And yeah, this is my 3. The other thing you need to be careful with while doing karyotyping, it's don't put your chromosome upside down, because in the lab, in the cytogenetic lab, everything counts. You see, what if there was a deletion there or translocation of a band or anything? You may not be able to pick it up. When I look at this one, I think of my 1. You see this fluffy thing made me think of my 1, so I will happily put it there. You see, the centromere is centrally located. Yeah. So yeah, what is this? This looks like my 5, but I think I'm 90% sure, 90 not 100. But so I will leave this guy here. If I change my mind, I know where to fetch it. This, when I look at it, I think of my 13. By the way, my natural nails grew to the length of my tip. So I keep on saying that I have to suck off the tip, but whenever I want to, this looks like my ex. Oh no, I'm not talking about ex relationship wise. I'm talking about X chromosome. Because I'm like, this looks like my ex. How can my ex looks like a chromosome? Yo, Nicole, come on. Be serious. That was a joke. But seriously, how can an X looks like a chromosome? Unless, I don't know. Well, chromosomes are cute though. This looks like my 3. So I will happily put it here. And then I have to double check just to make sure that I didn't invert anything at all. So this is my 16, definitely. It looks like the flower, but with the extra bands. Ah, you, come here. See, this chromosome likes to party. It looks like a flower. Long nails. It looks like a flower, but with the extra band close to the thing, to the centromere. So I will happily put it here. Okay. So this is giving me 18 vibe. This is my 18. I'm not having a vibe anymore. I made a choice. This is my chromosome 18. I will happily put it here. And I'm gonna come back. If you are having a second thought, which is normal, you are human, you can always come back. Place first those ones that you are so certain of, to save time, because in the lab, you know, we are very busy. We don't have time to waste. So you may want to, you know, save time. So I'm gonna keep placing them in where they belong. I used to say when I'm looking for the right spot for a chromosome, I would say, like, I'm looking for a home for this chromosome. So this is my 5. You can see the space. Oof, I have a problem. I'm not happy with this. This is my 2. What is it doing at my thing? You see this empty space and lots of bands. What was it doing at my 5 section? That's why I was like, I'm not happy with you. This looks like my 8 because of this fluffy, you know, top of the P arm, and then this dark band that tries to pop. But I'm gonna put it here so long If I change my mind, I'll let you know, but it does look like. This looks like my 11 because the PM is bigger than 12, and then this dark big prominent band, it's lower than that of the 12. So I'm gonna put it here under 11. This is my other how do you call this, 16, but if I change my mind, I'll let you know the changes that I made. You are actually doing this with me. This looks like my X chromosome. I have a feeling this patient is a female, so yeah, let's continue. This is my 10 because of this dark band here underneath the centromere that pops. I'm gonna put it here. This is my other 7. You see, because of this dark band on top there that is close to the edges, I'm gonna put it here and here. So this is my chromosome 15. I'm going to put it here. Yeah. Okay. We are almost done. Oh, this is my chromosome 6. You see the window that we have on the PM? Chromosome 6, I'll put it here. This is my other chromosome 6, but the difference is just that it's bent. You see, but you can see the window as well. Yeah. So I'm gonna put it here. Okay, this is chromosome 19. The flower, remember, the centromere is very dark. This looks like my chromosome 8. See this fluffy, and then here are these dark bands that just makes it obvious that it is chromosome 8. So what I'm gonna do is I'm going to put it close to the chromosome 8 that I had before here. Okay, yeah. So this is my 17, my chromosome 17. This is my chromosome 10. I'm gonna put it here. This is my 19, I think. Let me finish. Yeah. So here with our, okay, we have our one of our 9, now one of our 10 and 12 that we need. Where is my other 5? This looks like my other 11, so I'm gonna put it here. This is my other 13. You see the thing, the dark band. the two prominent dark band at the bottom of this acrocentric chromosome. So I'm gonna put it here. Okay, we almost done. Gosh. This looks like 15. This is my chromosome 15. I'm gonna put it here. This is my chromosome 18. I'm gonna put it here. This is my chromosome 14. I'm gonna put it here. Okay, this is my others. Not so sure. Looks like my 20, but I'll get to that now. This is my 21. I'm gonna put it here. These two, I will get to them now. I just need this, but this is my 22. I'm going to put it here. I have a feeling that I have missing chromosomes, but ooh, there's another 20 here. There's another one here. I think this is my 5 that I've been wondering in silence. She didn't want to worry you, so I didn't tell you, like that I was worried that I couldn't find my 5 when I didn't have any submetacentric chromosome anymore. I only had like acrocentric chromosome that I like left unplaced on my thing. Okay, let's check this one. This is my other 14. I'm gonna put it here. This is this is my other 17. You see the band, yeah. I'm going to put this one here. Yeah, so my acrocentric are complete. And I need my 20. Where are my 20, please? I think they are up there. This is my 21. You see the dark prominent band, yeah. This is my 21. What are you doing here? This is 22. This is 21. See the prominent dark band here. So this is 21. I'm gonna put it here as well. So this is my 20. Oops, sorry. Why? Because you see these dark bands on top here. Can you see? I don't know if this camera can focus. It's quite prominent. So this is my 20. I'm going to put it where is that? I'm gonna put it here. What is this one? I don't know what this one is. This is my X chromosome, because X's are not chromosomes. That is a joke. Okay, I'm gonna put it here. So I'm missing my chromosome 9 and 12. I am so worried. Where's my chromosome 12? Okay, this is my other 19. Where did my chromosome go? Do you know when we do this, we must be very careful, because chromosome can disappear and then it's going to compromise. I have a feeling that it's going to compromise your karyotyping. You must be very careful when you're doing your karyotyping. No chromosome shall miss, because you're going to get the wrong diagnosis and the lab cannot afford that. This is my 9. You see the long neck here, and the two bands are close to each other, which is different from chromosome from our sex chromosome X. So I am worried that my 12 is missing. I don't know if you noticed that. I'm wondering, like, where is it? Because I didn't move. This was overlapping, I think. That's it. Okay, let's look for chromosome 12. This is what we have so far. Can you see all the chromosomes? Okay, 1 is 1, 2 correct, 1 is correct, 2 is correct, 3 hold on a second. I had a feeling that I did invert my 3. What do you think now? Hold on. Okay, let me come back to it. 4 is correct, 5 is correct. Yeah, must be close to each other. 6, 7, 8, 9, 10 is correct, 11 is correct, 12 it is, X is 13, 14, 15, 16, 17, 18, 19, 20, 21. My 12, chromosome 12 is missing. We need to find it. Otherwise, my karyotyping will be incomplete. Where is it? You see, it's very tricky with this karyotyping thing. Ooh, I find it. I'm so relieved. It's so tricky. Once you lose one of your chromosomes, that's it. You mess the whole thing, because you're gonna count 45 when the chromosome are 46. When you count 45, you start thinking about monosomy. You start, if it's an X chromosome that is missing, you start telling people that you have Turner syndrome situation going on. You have a monosomy situation going on, when it's actually not a monosomy. The problem is you lost your chromosome, and then it's a mess. Okay, I know I've been talking. Where is it again? It's here. So yeah, we're gonna do this. We're just gonna put it here. Yeah, now I'm so happy because everything is complete. So we can count now: 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30, 32, 34, 36, 38, 40, 42, 44, 46, with 0 Y chromosome, which means what? This patient is not a male. This patient is a female. Why? Because we have 2 X chromosome here. So our ICN number or ICN will be 46. Why am I using a pencil? After such hard work, I deserve to write with confidence. So I'm gonna do this. I'm gonna write our ICN. I usually do this. I write 4, 6, then I will do comma, then I do XX. Then I will sell normal patient. But you know it's a female patient. So there's no sign of deletion here or translocation or anything. So this is a normal patient. It is good for the patient because she's normal, she's healthy, she's fine. But for us, cytogenesis, it's not okay. We need some abnormality to score. You know, we study for years to detect these genetic abnormalities. We live for these genetic abnormalities. We can't afford not having some trisomies, you know, some Turner syndrome, some Klinefelter syndrome. We need them in the lab. So guys, this is pretty much the end. I'm so happy that you made it this far. I hope you learned something. Of course I do know that you learned something. Please subscribe, join the family, comment, let me know if you would like to see any other video as such. The channel has so many other video interesting, lots of sit down motivational videos about relationships. Everything is being shared that you may watch. Also we have fashion videos and all of that, so I'm everywhere. I'm just, you know, you can watch. And yeah, so comment in the section in the comment section down below and let me know what you think. And yeah, so this is how you do your karyotyping, and I hope that you are successful. Thank you so much. If you haven't yet watched my other videos where we drew the chromosome, I explained, and where I introduced the chromosome. So I hope these videos, because this is part 3, you can check part 1 and 2. I hope these videos helps you to better understand your chromosome and do your karyotyping successfully. Diagnose your patient with precision, accuracy, and great results. So thank you so much. And thank you to everyone who trained me. So I'm not so sure if I'm allowed to mention the company, but thank you. If you have trained me, you know yourself. Thank you so much. I appreciate it. I can see today with my chromosomes and karyotyping. So, okay. Thank you. I will see you in those videos. Bye. Sorry that you can't see me. Bye. See me in other videos.